What's up, everybody? Welcome to Logical, Plausible, Probable. We're kicking off the year with an interview that I've been wanting to have for almost 2 years, and I'm so excited to have Dr. Sy Garte joining us to discuss one of his most recent papers on one of my favorite topics, and that is abiogenesis. Dr. Garte, thanks so much for your time and being willing to explain some of the very important facets of the abiogenesis problem, as well as the continuity principle and the evolution of replication fidelity. It's going to be awesome. Thank you so much for your time. Appreciate you joining us. How are you doing today?
I'm doing well. Thanks. Thanks for having me on. I appreciate it.
Well, I'm very honored, and I know you had a late night over on Modern Day Debate with on this conversation, so I appreciate you doing back-to-back combos here on YouTube with all of us degenerate laypersons looking for new knowledge. But now, before we get into our conversation, I want to kind of set things up for people viewing us for the first time, and for those of you who are going to be desperately looking for ways to debunk all the things that we're going to be talking about today, and if what you're hearing doesn't actually conform with your worldview. So let me give you a little background on Dr. Garte. He was actually raised in a very militant atheist family, and professionally became a respected research biochemist, and he had a anti-theistic worldview. So he wasn't a theist. He wasn't all convinced by his God of the gaps brainwashing and such and cognitive bias. He actually came from the totally opposite perspective, and he had no intention of seeking God, the God that he didn't believe in. However, over time, the very science that he loved led him down the path and began to question the validity of the atheistic worldview. And ultimately, the Holy Spirit guided him to belief in God and Jesus Christ as his Redeemer. Now, I've had multiple conversations with Dr. Garte, and he's an extraordinarily humble man, but I strongly recommend, for those who are going to question what he's going to talk about today, if you actually go take the time to look him up, you will see that his H-index indicates that his peers view him as a credible source. And this has also led to him being tenured at not one, but 2 different universities, Rutgers being one of a very notable one of them. And he's also worked at the NIH. He's been editor of a peer-reviewed journal, reviews papers, all sorts of different things. And these are just a few of his professional accomplishments. So don't make sure you're listening to the things that he is saying, and he's saying them from an expert level, and don't just dismiss things that might not conform with what your talking points. And one last thing before we get into the conversation, he's also written a very incredible book. I really enjoyed it, about his journey from being an atheist to being a believer in Christ. It's called The Works of His Hands, A Scientist's Journey from Atheism to Faith. And I strongly recommend you check it out. I've got a link to it in the description, and it's also pinned in the side chat. Make sure you click on that and go get his book and really take the time to analyze things from his perspective, and as well as it really conforms with a lot of the atheist transitions, if you will. So anyway, enough of me rambling. Dr. Garte, let's get into your very interesting paper on the continuity principle and the evolution of replication fidelity. And to get started, can you kind of give us a quick background on what you got so interested in this topic as a professional, and all the way into, you kind of came out of semi-retirement to write a paper on it. I think you've done another one since then. And can you kind of explain to the audience what is being addressed, and what is this continuity principle versus the saltation differentiation?
Okay. Yeah. So first, let me just talk about retirement. When I say I retired, which I did from NIH, there's an old saying that scientists never really retire; they just do something different, and that's pretty much what I did. As a lifelong scientist, I never thought that I would actually stop doing science. I can't do lab work anymore. I don't have a lab, but I can do theoretical work. And in fact, I've always loved the idea of theoretical biology. I was never able to engage in it much, just lack of time, and I was too involved in other things. But now that I am retired, I can do it, and I've been doing it. I do have a few papers from my main career where I did do some theoretical work on, for example, on fractals and other things like that. But I had the chance the last 5 years to really look at the things that interest me from a theoretical aspect. And one of the things that interests me a lot is this whole question of abiogenesis. And what led directly to this paper was my curiosity about the fact that when in all of life that we know, and we'll get to this a little later in detail, cells have to replicate themselves. And that whole fact is astonishing, because there's nothing else in the universe that replicates itself, not with the high accuracy that cells do. And as we'll see, that high accuracy is required. If you have a low accuracy self-replication, let's say only 50 or 60% of the cell is replicated accurately. By that, I mean all the enzymes, all the components of the cell. If there are a lot of mistakes, cells die. This is what's called an error catastrophe, which we'll get to also. So I wondered, and so since very, very accurate self-replication is required for life, it's also required for evolution. And I wondered how such a high level of self-replication accuracy could evolve. If all the mechanisms for self-replication, like the genetic code, the ribosomes, the production of proteins, that's how a cell replicates. The only thing that can replicate in a cell directly is the DNA. So it's the DNA that then causes the accurate replication of everything else. All the systems, all the proteins, all the enzymes, and that's done by the translation system, which is incredibly accurate also. And that translation system is so complicated, I wondered, how did that evolve? And it dawned on me, maybe there's a problem here. And I decided to take a theoretical and statistical and systems-based approach. And you're much more of an expert on the kind of programming that I have to use for this, so I may even defer to you with some of the questions. But I was able to come up with a program that ignored all the biochemical details. Okay, it just said you have a cell. This is a very simple model. The cell is going to divide and replicate itself, and it starts with a certain fitness or a probability of survival, and if the replication is accurate, let's say it's 100% accurate, then both daughter cells will inherit exactly the same probability of survival. If it's not 100% accurate, there's a probability that some of the cells, some of the descendants of the original cell, will have a different value of that p value, the probability of survival, which we call p. I call p. P sub s or p. And it's that p value that determines whether the cell lives or dies. So it's a very simple model. It doesn't make any assumptions about whether RNA or DNA or something else was the genetic material. It makes no other assumptions about mechanism. It's just a statistical model to look at those two parameters. How are they related to survival? And for survival, I use the growth curve. So the growth constant, which tells you if the growth constant is positive, it says that cells are alive. If it's negative, then they're dead. So yeah, these are, I've already mentioned error catastrophe. Replication fidelity is how well does the cell replicate itself? In other words, to what percentage is everything replicated accurately? And that varies from 0 to 1. It's like a probability. The survival probability is also a probability, which varies from 0 to 1. 0 means the cell dies or has no chance of survival. 1 means it's going to live no matter what. And of course, there's everything in between. Now, let me just say this, which I think is not clear immediately, is that in modern cells, in modern organisms, both of those values are very close to 1. The replication fidelity of all living organisms is extremely high. It's like 0.999, 6 nines, I guess. So it's 99.9999% accurate. It has to be, and we can get into why when we start talking about some of your questions, but it has to be that high. If it's any lower than that, cells die. The survival probability is also pretty high. Of course, it's not 1 because death occurs, but it's very close to 1. So the best way to think of this is not us or animals, but single cell bacteria, for example. That makes it simple. And when everything is going well, bacteria do well. They survive. They don't die. They reproduce very easily. Their growth rate is very high. When a bacteria divides in 2 and both of those daughter cells are merrily eating away until they begin to divide, the growth rate is basically 2. In other words, it doubles every generation. That's the maximum growth rate you can get with a bacterial cell. And
quick interjection, Dr. Garte.
Sure.
If I remember correctly as well, for anybody who's looking at the paper, the growth rate you're referring to, I think in the formulas that's represented by K, and in some instances R, right?
Yeah, and one of them is the log of the other. R is the log of K. So for K, which is more easily calculable, the 1 is the borderline between life and death. So anything less than a K of less than 1 is, in other words, a negative value of K implies eventual death, because instead of growing, the cell population gets smaller and smaller with every generation. But if the K is greater than 1, then the cell population continues to grow and does not die out. So what you want is a growth rate greater than 1, and R is the log of that. So that's why you get 0 whenever it's 1. So 0 is the border between life and death. So that's the growth rate story. And in this paper, I used K because, as I said, it's easier to just get directly from these other parameters. And what I wanted to do was see what is the what is the relationship between growth and these two other parameters, survival probability P and replication fidelity F, and is there a relationship between those two? And I found out that there is, and that's listed in the paper. And the other thing I wanted to test was the whole issue of continuity in evolution. Continuity is sometimes called gradualism. It was a Darwinian idea. It's one of the things where Darwin was wrong, because he said if anybody can show that gradualism is false, my theory falls apart. But that's not true. We know several examples of discontinuity. For example, the origin of eukaryotes, which was a single event. The origin of vertebrates was an event, one event, a molecular event, which was whole genome duplication. So discontinuity occurs during the evolution of life. It's rare, and it needs an explanation whenever it does occur. So what I wanted to see was, how does continuity apply to the origin of life? Because the naturalistic ideas, I wouldn't even say theories, because there are no real theories, but the most people think of the origin of life, by most people I mean most materialistic scientists who are studying this, think of it as a gradual process like everything else. And they like to say that eventually life evolved. But when they say evolved, even they know that it's not biological evolution, it's chemical evolution. So again, they pose a stepwise process. First we had RNA, and then RNA became DNA, and then there was replication, which got better and better, and eventually replication was great. And so my question was, is that feasible? Is it really true that you can start with a not very good cell or not very good strand of RNA, and it gets better by evolution in a gradual process? And what I found was pretty clear. The answer is no. Not if you start from a very low level, or even from a moderately low level. You have to start from a level of both probability survival and replication fidelity that's at least 60%. Now, that's a very low level in terms of modern life. But if we're talking about early life, we're expecting that the survival of early life forms, whatever they were, was probably pretty low. And why would their replication fidelity be high? Because after all, it's unlikely they had the complex systems that we have today. They probably didn't have error correction. They didn't have, any of the things that we now have. So you'd expect a moderately low level of fidelity. And what I found is that there are thresholds. If you get below a certain level for both of those parameters, nothing happens. You can't evolve. You can't make it better because the cells die out.
Interesting. So
so Jim Tour always says time is not on the side of abiogenesis. Time is the enemy of abiogenesis, and that's exactly what I found. Because if you have a K value that's too low, every generation makes things worse. You keep losing population. You see?
Well, and correct me if I'm wrong, but in a lot of the research, even when they are getting things to supposedly self-replicate to some extent, they are seeing a loss over time, like over cycles. They end up seeing a loss They get in some, to dumb it down, the sequence literally gets shorter, that kind of thing over time.
Yeah, I mean, I don't want to go as far as Jim often does, but frankly, the abiogenesis literature doesn't tell you the whole story. Right. I mean, they don't tell you all the things that don't work. And what you just said is right. I mean, you can find it in the literature if you look hard enough. But the whole idea of RNA self-replication, that is not actual self-replication. Gerald Joyce uses that term, but it's not. It's a different. I don't know why he uses it. What he's really talking about is ligation, which is different from self-replication based on a template. So,
It's interesting. It's interesting. I want to tell everybody, you need to go and you and I had a conversation about this, but you need to go dive into the paper that Dr. Garte's referring to, because earlier today, I actually had a conversation with a doctor, like a medical doctor, and he was going on and on about exactly what you're talking about right now on Joyce's, the replication and whatnot. And I'm like, no, it's a ligation.
Right.
And he insisted that I was wrong and that had been accounted for in the most recent papers from Joyce, all this kind of stuff. And I'm like, no, it has not. This has not been accounted for. It's they've made it a little bit more complicated, a little bit more advanced, but they still haven't solved the problem. And but it's really amazing how folks don't get it. So,
this is not deliberate prevarication on Joyce's part. He admits this readily. I don't know why he chose to use the term replication. It confused everybody. And in other things of his, I've used in other shows. He makes it very clear that he agrees with everybody else that there's a long road to go, and it's not looking good. So anyway, yeah.
Well, and I think kind of to correlate with what you're saying is I know one of the things you addressed in, pretty early on, you said, I'll quote you here, the requirement for minimal replication fidelity to allow for survival and evolution in early cells in any hypothesized RNA world has been estimated at approximately 0.98. And that was from Shostak, which is a very well-respected origin of life researcher, right? But scenarios for the evolution of replication fidelity and information molecules are unknown. And we've been kind of talking about this in relation to abiogenesis, but that 0.98, I mean, am I correct in this that if the modern extant organs fidelity we're observing is 0.9999999, right? So is Shostak saying, well, you basically need to be at 0.98?
No.
What's the difference in what he's saying there?
The difference is okay. So it's very interesting. There's a very simple formula to determine what is the minimum mutation, or the minimum replication fidelity that you need for a viable cell. And it's simply 1 over the length of the genetic material. So what he was talking about was the barest minimum size of RNA that will allow for life. And he was using 500 nucleotides. No, sorry, 50 nucleotides. Okay. And 1 over 50 is 0.02. So that's a 2% error, which is a 98% replication fidelity. But that's for 50 nucleotides. There's no gene that small. That's based on the theoretical hope that a 50 nucleotide RNA might code for something. The most it could code for would be a tiny peptide, like 50 divided by 3,
So in line with that, at that point, obviously what you were referring to earlier in relation to the error correction mechanisms, all the proteins that are handling all of the fidelity that we observe,
right?
Basically, it's not even remotely plausible that a 50 nucleotide sequence is going to be able to account for that. Because correct me if I'm wrong, but some of those are 1,000 plus. I mean, you're getting into a lot of a minimum 300 usually. Sorry, I mean, and there's multiple working together in unison. Is that a fair kind of?
Yeah. I mean, absolutely. And what 50 nucleotides could do would code for a 20 amino acid peptide, which that's not going to be a catalyst. That's not going to be an enzyme. I don't know what that'll do. What it might do is it might help RNA replicate. That was Shostak's theory. And he did publish a paper showing that peptides can, in fact, help RNA replicate by avoiding the self-annealing problem. But that paper was retracted because it turned out to be a mistake in the lab. And so at that point, he has the whole RNA world hypothesis is in deep trouble at this moment, because it doesn't look like RNA has the potential to serve as the genetic molecule in any meaningful way. And we can get into that more a little later, because I do mention RNA World a bit in the paper, and I can explain that.
Well, I know from some of our previous conversations that RNA World's kind of it's on its way out, and
yeah, it is.
There's some new hypotheses that are coming in, and it's kind of funny how so many folks, especially more in the layman, and I guess even some of your peers, like that aren't reading the origin of life research, are still kind of thinking that RNA World is still what's going on, and it's like, no, we're kind of into some different stuff now. But even PhDs like yourself are. Again, if they're not reading the kind of papers and writing papers like what you're doing, they might not be aware of the fact that something like, oh, this was retracted. Like we haven't actually solved the annealing problem. And because I mean, a quick sidebar, can you kind of explain to the audience what the annealing problem is? Because when I found out about this, I was like, oh, okay, this is a serious problem.
Well, RNA is very good at base pairing, like DNA is. To itself, what to other things, and that's important for messenger RNA to do that with DNA so that it can serve as the message in the cell. It base pairs to the DNA sequence, and then it's messenger RNA, then it goes and it carries that message to the ribosome where it makes proteins. The problem is that RNA is too good at base pairing, and so if you have a strand of RNA that you're trying to get it to replicate itself, what it'll do is it'll bind to itself. That's called self-annealing. It folds over and it binds to itself. And then you're stuck because it's not going to let go. I mean, in modern life, there are all kinds of mechanisms to prevent that and to get the RNA off the DNA once it's stuck. Because there are enzymes to do that. I mean, as you know, everything happens by proteins and enzymes. And what's interesting is when I wrote the paper, I mentioned RNA World as just one of several possibilities, and one of the reviewers chastised me. And this is not a theistic reviewer. This is a mainstream regular journal. And one of the reviewers said, well, you shouldn't be mentioning RNA World because it's almost definitely not in contention. And he then told me, he or she then included several references, which I used in the paper, which I had not been aware of, attacking RNA World as a viable hypothesis. And that was several months ago, almost a year ago. And it's only gotten worse since then. So RNA World by itself has really given way, as you said, and now it's RNA Peptide World, and I don't know, that's not going to last very long either because there's just not enough evidence for it. So there we are. It's sad. I mean, there really are no theories for abiogenesis that go anywhere.
Well, yeah, I mean, it's interesting how like, oh, this one accounts for like these 3 problems or this one problem, but then ignores the other 7. And it's like, oh, this one I counted for this one, but it's like but you had to willfully ignore the barriers on the other side of the equation in order to solve this one. That's I find that it's very interesting to me how like that seems to just either go over the heads of a lot of people or be willfully ignored by many that are aware of the problems. Like, oh, we'll solve that one at some point. And I'm like, so it doesn't matter. It's like, cool, you figured out how to make X, but you can't do it in this scenario. So if you needed to operate in both scenarios, you have to account for both of them simultaneously.
Yeah, exactly.
I mean, it's just that simple. Like, and to me, when you really contemplate like things that humans do on a daily basis, like we freely recognize that, like, unless I have the screw and the screwdriver, I'm not tightening the screw, like, or at a minimum have something that replicates the function of the screwdriver, right? Yeah. But then you also have to account for like, how did I make the screw in the first place? And as you alluded to, like Dr. Tour kind of references this. It kind of cracks me up how wannabe Dave, as I call him, missed the whole point that Dr. Tour was making in like, oh, basically nobody even knows how to put all the car parts together. However, with abiogenesis, we're talking about how did you make all of the parts, not how did you put them together. How did you make the parts is the point. And so many, and this obviously in the context of your paper, it's all about like, hey, how do we account for the survivability and the fidelity simultaneously? And
and that's one of the reasons I didn't mention RNA or protein or DNA in the paper, because it doesn't matter what the actual parts are. What matters is, can you theoretically get a living population of cells that continues to increase in size with a positive K value when you don't have a high probability of survival or a high probability of replication? And let me go back to this replication probability. You asked earlier, why is it that modern life has to have such a high level, such a low error rate? And the answer is that formula 1 over L, because L for us is in the billions. So it's 1 over 10 to the 9th. And that is the mutation rate for all animals, is 1 in 10 to the 9th errors.
Okay, I know you.
So that's incredibly low. It implies this huge. That's for DNA replication, and then you add in the error for protein synthesis, which you also need in order to have replication of the phenotype, and that also is extremely low. So the two together, if you multiply them together, it's 99.9999%.
You're having to combine the probabilities, what you're saying, in terms of
because because they both have some errors, so you multiply them and you get the final error.
Okay, so let me ask you a question. So in living cells, we have massive discontinuities that are writing from basic, even a single mutation, right? I know you had, correct me if I'm wrong on this, but I think you professionally were involved in cancer research, weren't you?
Yes, I was. Mm-hmm.
Can you kind of put that for the audience? Can you kind of put what we're talking about in terms of the fidelity necessity? Can you kind of put that in context of like a cancer that would be caused by X type of error? Does that make sense?
That's interesting. Yeah. So cancer is a genetic disease. It starts with the genes. It is a error in some genetic sequence that makes where the protein that's made has now changed, and it does something either too much or too little. I mean, it's very hard to generalize because there are many different pathways. So now you would think that would be a problem for evolution, because there are errors all the time. But the thing about cancer is that it doesn't affect. It takes a very long time to develop, because a lethal cancer is not just a single mutation. That's the beginning of it. But then there's a long process. In humans, it takes about 20 years from the time, for example, you're first exposed to a carcinogen, asbestos or whatever, to the time you actually develop the disease. In animals, it's half the lifespan often. So cancer does not affect growth rates, because it doesn't kill. It kills basically older animals, including us. I mean, cancer is much more prevalent among the old. So it has no evolutionary significance. Okay, it doesn't come into evolution because it doesn't affect reproduction. And the interesting thing about cancer is, when cancer cells become very advanced, they're continually mutating. Their fidelity becomes lower and lower. What they do is the same thing that bacteria do when they're threatened, or viruses, which we now know we're right in the middle of. When they're threatened, bacteria will have what's called hypermutagenesis. They mutate at a huge rate. Their F value goes way down, and they mutate. Most of them die, but a few will survive, and that keeps the population going. It's exactly what happens with a virus like coronavirus or any other virus. Viruses are the only exception to this 1 over L rule. They have a mutation rate that's extremely high, way beyond any form of life. And the reason is, they don't care whether they live or die. By care, I mean it doesn't come into play. Viruses mutate all the time because that's their strategy for long-term survival, because eventually you'll get a Delta or you'll get an Omicron, which now can live better, can transmit better, can be more infectious, etc. That happens with every virus. Happens with the flu. It happened with SARS. You get this. What we now have with Omicron. Why does Omicron only affect the upper respiratory tract and not the lung? Well, it's milder, but it's also more infectious, and that's what a virus cares about, being more infectious. So that's evolution. That's evolution in practice. This is well known. And what it says is that you can get away from this limit of error catastrophe if you need to. And people have talked about this error catastrophe in the context of antiviral agents, things that will increase the mutation rate even further. So eventually the viruses just die. And that also happens in nature. We'll get a little off the topic, but that's an interesting and important question, is this issue of, why is the mutation rate what it is? And in normal life, and I'm talking about from bacteria all the way up to us, it's extremely low. Otherwise, you just get massive death because proteins stop working.
Gotcha. Yeah, that makes that definitely makes sense. Okay, so let me skip up here, and I'm going to read this because I don't want to try and butcher this too much. Okay, so you got into some very interesting stuff here, and yeah, but for folks who are reading through the paper, this is in the section Evidence for Discontinuity in the Evolution of Replication Fidelity at Low Survival Probability. So that's a mouthful. And you look at a variety of scenarios based on different factors. You kind of alluded to this earlier. And at the end of this section, you said equations 5 and 6a suggest even with perfect replication fidelity, F sub r equals 1, values of p below 0.625 will give k less than 1 and lead to extinction. This result was confirmed by the experimental simulations, which consistently gave a value of p sub s between 0.63 and 0.65 as the minimal value of p to allow for population survival and evolution. Can you kind of what does that mean? And is my assumption that even allowing for fidelity significantly less than what we see in modern organisms, there appears to be a very hard limit, as you kind of alluded to, in relation to evolution, like plausibility, I guess?
Yeah, well, this is just sort of a theoretical glance to see which is more important, the fidelity of replication or the survival probability. It turned out that, contrary to what I expected, I thought replication fidelity would be the most important thing, but it turns out that survival probability is actually more important. So you could have actually perfect replication fidelity, which is not likely, because why would you have that? But even if you did have it, if your survival probability goes below that value, the data show that, that's it. The population is going to die out. And that's interesting, because what that suggests, that explains why bacteria, for example, who are doing perfectly fine, they're replicating very well, but now their probability of survival goes down because they've just been exposed to an antibiotic or some other toxin. And if it goes below 65%, they can't recover. And that's why bacteria die with antibiotic. If you have an infection and you inject antibiotic, you're knocking down the PS way below 0.65, who knows what. And as long as you're below that, it doesn't matter how well they replicate. They're replicating a low probability of survival, and they die. So, and in fact, and I wasn't going to talk about this, and I won't, I'll just mention very quickly that I have started using the same program to examine how hypermutation works, and it works, and the program works very well, and that's going to be another paper, which I'm still writing. I'm still in the middle of. So anyway, I like the model. It works well, and I was surprised that survival is more important than replication, but okay, that's what the data show. But the point is, what you said is correct. How do you get simultaneously a high probability of survival, a high survival probability, and a high replication fidelity from scratch? Okay, it's not that one of them can be continuous and the other not. That neither one is continuous. So if you start with, 0.4, for example, 40% of replication fidelity or 40% of survival probability, you're done. You don't go anywhere.
Yeah, that and obviously you're not even this is one of the things that really stuck out to me was you're not even talking about getting to 51. You've got to get to 60.
Yeah, it's above that.
Right?
65.
And it's not even passing the 50-50 mark, which, I mean, heck, passing 50-50 is tough.
Oh yeah, no, it
would be. I'm saying like even like going from 0 to 51 is a leap, right? And then it's like, no, we actually have to go. It's not just getting to 51. You got to get to 65. In that 60. That's a whole nother additional like in terms of the gap, if you will. Like you have the chasm that must be crossed and all this stuff. And I think a lot of times people don't really sit down and contemplate like, yeah, this is numbers and all these kind of things. But when we're talking about like real life deal, the point you alluded to about, what Dr. Tor says, time is the enemy. Like you've got to get this at the same time. At some point, even if you want to say, oh, it took X number of years to happen, at some point in that time frame, you had to have accounted for all this happening simultaneously. You can't go back into this one happened first and then this one. No. At some point, in time, they have to physically exist, not only at the same moment in time, but also in the physical proximity to each other, literally next to each other from a molecular level. Like there is no way around this. And I think very often people kind of forget about that. Like, oh, well, no, there was, all these simultaneous actions were happening in all these different places around the world. I'm like, I don't care if problem 1 gets solved in Australia and problem 2 gets solved in the Antarctica. It doesn't matter. Like,
that's right.
And correct me if I'm wrong on this, but from a molecular perspective, it's the equivalent of, being, the equivalent of, 2 dimes sitting next to each other on the table. And from a molecular perspective is the equivalent. If molecule or the action in molecule or dime number 1, cool. Action number 2 is in, the other dime, and they're sitting next to each other. That's the equivalent as a human traversing a exorbitant distance, from a size perspective, right? Like, so it doesn't matter if, cool, I did this in the United States and the other things, and the store I'm going to buy something from is in England. I still have to get across the Atlantic Ocean, and you have to have the plane or a boat to make this happen, right? And it seems like so many those aspects just get completely glossed over. Nobody wants to actually analyze what you're showing to be, must be accounted for from a research and a probabilities perspective. You've done the math, you've done the simulations, and the simulations are saying, hey, you must account for this.
Right. Yeah, exactly. You're right. I mean, I have nothing to add to that.
Now, you also kind of in the next section, folks, but again, this is a very interesting paper. It's worthwhile to take. I read it like 4 times to make sure I kind of understood what he was talking about. So you also allowed in some of the simulations for different initial conditions and for both beneficial and deleterious mutations. And I think there were some pretty interesting results. So can you kind of explain to us what you did by adding them in? And also, if I remember correctly, you had a 1 to 20 beneficial to deleterious ratio. Is that right?
Yeah, I used different ratios. In fact, in later work, which is in that other paper, which just came out, I have a second paper where I go into much more detail about the methodology that I used, and I talk about the effects of this ratio of beneficial to, because people could say, well, in early life, mutations were probably less deleterious than they are now. In other words, the balance between beneficial and deleterious mutations has probably gone down with time, because now we have a very good system. So most mutations nowadays in modern life are either neutral, they don't have any effect, or they're deleterious. The fraction of mutations that are beneficial are tiny. And usually a beneficial mutation starts out as a neutral mutation and it just stays, doesn't do anything. And then the environment changes and all of a sudden that mutation allows the person who has it or the animal who has it to do better, okay? But in early life, whatever kind of molecule is being mutated, and of course I make no assumptions, but in early life, one could imagine that if early life really did start as not very good, in other words, not very successful, with a low probability of survival, then you can imagine that mutations might help more than they would hurt. And so I looked at the effect of that ratio from 50, which is roughly what it is today. That's from the literature. 50 deleterious to every good beneficial one, all the way down to more beneficial than deleterious, which sounds crazy, but I wanted to do it just to check. And so I had the final ratio was something like 0.2 deleterious. In other words, one beneficial for one deleterious for every 5 beneficial mutations. So many more beneficial mutations. And it made very little difference in the model, which I was surprised to see. So I ended up using 20 as a sort of intermediate, because between 50 and 10, there was almost no difference at all in the results. There was a small difference under 10, but those values are not realistic. And when I got under, when the ratio was less than 1, in other words, they were more beneficial to deleterious, a lot of things changed that made the model look kind of crazy. So, but it didn't have any effect. So on the final result, on the value of K, so I said, okay, we'll just stick with 20 to 1. Didn't matter. And the key, the most important parameters were P and F to define the K value in these simulations.
Well, kind of in line with what you've been kind of mentioning here, there's a portion, some of you said in the papers, 2 different things. Let's see if I can find this real quick. I think it's very often overlooked. You said, quote, in modern life, there is a threshold of replication fidelity below which cells cannot survive. At high mutation rates, which can be as low as 0.1 or F is 0.9, modern organisms undergo an error catastrophe from which they cannot recover. And then in the following paragraph, you say, in complex modern cells, survival probability and replication fidelity are linked, so that deficits in replication fidelity at either the DNA replication, transcription, or translation phases results in decreased P in future generations. So am I correct in my understanding of this, that without the high level of fidelity we currently observe, then not only would survival probability collapse, but that even if replication occurs, the future generations would have lower and lower probabilities of survival, potentially all the way to extinction without somehow regaining that higher fidelity. Is that correct?
Yeah, that's right. Well, I mean, it gets complicated because usually that is correct, because I mean, if your p-value is low, that means you're going to have a lot of mutations. I'm sorry, you're going to have a lot of death, because death is 1 minus p. Okay, so probability of survival is 1 minus the probability of dying. So if your probability is low, it's likely, let's say it's less than 50%, it means that more than half of the cells will simply die because they're not metabolizing well, they can't get energy, something about them doesn't work that great. So they have a high probability of simply dying. And that applies also to the ones with very high replication fidelity. They'll replicate nicely, but if their P is low, they're just going to die anyway. Okay, so what I was talking about, modern cells, see the combination of P and F is what evolutionary biologists refer to as fitness. Okay, how likely is it for an organism to live, to survive, and how likely is it for it to pass its inheritance onto future generations, and that is basically replication fidelity. And those two are linked in what's called fitness. And the reason they're linked in modern cells is because one of the things that makes cells more likely to survive is to have a high replication fidelity to allow cells in the future to survive. So it's a little bit of a complex, twisted sentence. I probably could have written it better, but I didn't. Right now, I didn't say it much better, so maybe not. But what I'm trying to put in there is people who say, well, where does fitness come in? That's where fitness comes in. Okay, so the overall fitness of its population is based on both of those parameters, which are highly linked in modern life.
Well, Dr. Gar, are you suggesting that the variables that are necessary for life to exist are complicated? that there may be a few things that must be accounted for that aren't just easy to like, oh yeah, we'll figure that out later. Like, no, you gotta account for these things in the front end. Now, one of the things I thought that was very fascinating was in the simulation, you remove some of the requirements, and this is where I thought things got pretty interesting. So you said the simulation data show that at higher levels of P, the evolution of efficient and accurate replication can proceed with a high degree of continuity throughout the entire range of initial replication fidelity. However, the converse does not hold true. For low initial P values, less than 0.4, the evolution of increased replication fidelity requires large jumps in F sub R that suggest a salutation evolutionary path. Furthermore, there is no continuity from low P to higher values, depending on the simultaneous value of F, even at perfect replication fidelity, F equals 1. The minimum level of P must be at least 0.6 in order to allow for any possibility of growth, survival, and evolution. Regardless of the degree of replication accuracy and mutation rate, cell populations with initial values of P as less than the approximate 65% cannot survive. Now, we've been talking about this kind of overall, but can you kind of like explain to us again what that meant and then how you were executing that in your simulations?
Oh boy. Okay.
Oh
yeah. So
yeah. I'll follow up on that. I have another question, but yeah, go. And also, I think you, if I remember correctly, you also kind of alluded to, well, maybe we have to account, like, just accept this being analogous to Crick's frozen accident. Was were you like, was that a reference to like how do you get to the 65 level needed? Is that what you're talking about in terms of the frozen accident?
No. Well, in a way, but let me get to that. So yeah. So what I'm saying in that paragraph is basically what I've said before, that any way you look at it, you're going to have a discontinuity unless you have, by some amazing chance, and that's where the frozen accident comes in, the first cell life, the first cell comes with both a high level, defined as over a certain threshold, of survival and a high level of replication fidelity. And that's Crick's frozen accident idea was how he explained the evolution of the genetic code. Because the genetic code is similar to this in that it's very hard to imagine how it evolved, although people are working on that. But the difficulty in understanding code evolution is you can't change it very much. It's not like you can suddenly change, one of the bases and then life goes on as it was. See, and this relates to a very important issue in biology in general, which Dennis Noble has pointed out, is that when mutation occurs, it depends where and how long this mutation happens. Mutation is critical to things like the immune system, right? You get a lot of variability. But as Noble has pointed out, that variability is restricted to a very short part of the immune genes, the antibody genes. And the reason for that is that the rest of those genes, called the C region, the constant region, are required for antibody function. So if you let the entire gene for an antibody experience a high level of mutation, you might get all kinds of variation, but you'd also get everybody dead because the genes wouldn't work.
Well, and real quick in line with that, for folks, if you've never seen this, I strongly recommend you searching on YouTube, like how the antibody is formed and like all the different types of well, there's all sorts of aspects of your immune system, but like the point that Dr. Garte was making about these conserved regions, when you're actually talking about the physical three-dimensional structure, the conserved portion of the gene is like the foundation, like the structure and the arms, and then like the variable portion that Dr. Garte's talking about in terms of the portion of the sequence that gets can be more random is changing a fixed portion on a three-dimensional object
Right.
that now can be
very small.
a very small portion of it. So it's like you're like, is maybe a horrible analogy, but like you've got a socket set, right? And you've got the ratchet. So you've got all these different sockets you can attach to it and flip them in and out, but they all fit onto the same ratchet.
That's actually excellent. That's a great analogy.
And something really struck me about this. I don't know if you've seen these, Dr. Garte, but they got some new sockets now that one socket you can actually fit a plethora of different size nuts. And the way they did it is,
I have one.
You know, okay, awesome. So you know those universal sockets, right?
Yeah.
To me, it's kind of like the similar type of premise, right? You have one that can form all these different. It's not necessarily made for one size.
That's right.
And it can be flipped in and out. But at the end of the day, you're still attaching it to a ratchet.
Right.
And that portion remains the same. The overall size, shape of the socket remains the same, but you're being able to account for different variabilities. And I think a lot of times that randomness that you're referring to people think, oh, well, that just means, as you were kind of alluding to, like, oh, everything's just completely undirected and it's just going wild. And it's like, yeah, well, sort of. Nope. It's kind of like, it's controlled mutation, if you will, in that context.
Exactly.
To me, that's always kind of struck me as something that needs to be addressed, like controlled versus random. I mean, that's there's a controlled randomization, but that in and of itself is not random. Exactly. And it's something I think people need to think about. But anyway, okay, so
well, let me get back to this frozen accident story.
Right.
So that idea was interesting because he didn't have any other way to explain it, but it's kind of no longer believed as seriously possible that you could get a genetic code that just pops into existence. And popping into existence is not something that anybody studying these things believes. It's just too crazy. It's too, the probabilities are too low for that to happen. So why did I put that in there? I put it in there because I wanted to point out, in a way, I didn't actually say it this way, but I wanted to point out that the idea that this would happen is actually just as crazy as the frozen accident idea. So when I said it's reminiscent of this, in the background was my thought that everyone will say, yeah, that's not possible because nobody believes in the frozen accident.
Interesting. Okay, well that makes a lot of sense, and I'm assuming that that may have been kind of the kickstart point of kind of what you followed up with, like the next portion of what you started talking about, which I thought was very interesting. And this is something you and I have discussed. We kind of talked about it earlier in this conversation. Is that the RNA World Hypothesis is, it's just kind of starting to lose favor with the majority of the active origin of life researchers. You kind of lose this multiple times, but towards the end, I think it was in the discussion portion, you said, quote, given these conclusions, it would be profitable for origin of life researchers to focus on highly stable metabolic systems, regardless of their initial simplicity, rather than on the evolution of replication systems within early cells with questionable degrees of survival probability. So is this more kind of what you're willing to in terms of the frozen accident, or are you referring towards, like metabolism first ideas being more likely, or just that regardless of the concept, it must be highly stable in order to have plausibility?
Well, okay. Why did I put that in there? My goal was to publish this paper in what we would refer to as a mainstream scientific journal, peer-reviewed, etc. And I was successful at that. But to do that, there are certain things you can and cannot do. You cannot, in such a paper, refer to the hypothesis of creation or anything that sounds like that. And it's a good idea to say in the discussion how these results impact the field in general, if they do, and in what way. And what I'm saying there is that these results kind of suggest that focusing exclusively on the replicative first issue, which has gained a lot of favor recently, may not work, because what I'm talking about here is replicators. And so the alternative, of course, is metabolism first, which I used to be totally against, but now I'm in favor of. And I think a lot of evidence is coming out about metabolism first, some of this work by Jeremy England looking at physics and metabolic cycles and some other things like that. So it might be that would be more useful. But the fact of the matter is that I don't think either of those two work. Neither the metabolism first nor the replicator first are actually very good ideas. But I can't say that in a mainstream journal because it would have simply been rejected, to be honest.
Well, you know, I don't know if you read Jeremy England's book, his most recent one.
No.
But it was kind of interesting in that he freely states that there's like gigantic barriers.
Oh yeah.
Thermodynamic barriers to
sure.
Different things that he talks about, and then of course when he gets all the way up into beyond the normal age of abiogenesis stuff and gets into like the barrier between prokaryotes and eukaryotes. I think it was something ridiculous, like a 200,000 x thermodynamic barrier you had to account for or something. I forget the exact number. It was something around that level. Now, of course, he still thinks that there's ways to account. Hey, this could happen, right? And it's like, okay, fair enough. I mean, I guess. Well, yeah. But at the same time, I find it very interesting how folks like England freely admit that there's gigantic chasms even in their own hypotheses. Yeah.
And there always are. I mean, there certainly are in mine too. It's very simple. It's very simplistic. It doesn't take into account many complicated issues. But I find that actually as an advantage because it doesn't rely on any of these assumptions. This model would apply to any biochemical specificity of any life form. Right. It's the same thing that's still true.
Well, it's kind of interesting the point you were making earlier about the on the frozen accident side on the genetic code of you have major changes, you have death, and to me something as simplistic as like an AUG start codon. It seems to me like a lot of folks aren't really thinking about that in the context of if you have an error in the start codon, you have a serious. Like you got a serious problem. So like if all your your ribosomes are configured, let's assume hypothetically you somehow get this, like which in and of itself was a whole nother problem, right? But let's say you've got one. What happens if you have somehow an error in start that all these other components are programmed to recognize? And if you don't have that anymore, like you're toast. Like you're dead. You're immediately, right? Or relatively immediately. Yep. And you sure as heck aren't going to have replication fidelity of any way, shape, form, or fashion. And I don't know. I just always find those things, this is why I was so interested in your paper of just like reading through this and looking at the math. And as you said, removing all these hypothetical, assumptions from as many as possible from the equation. And then being like, well, here's where the rubber meets the road, if so to speak. And no, that really struck me. Now, there's sidebar, folks. If you want to, we're gonna have a very, very short Q&A. If you have questions, I know people are having a hard time with the spreadsheet that I spent. So I post another link inside chat. There's a link to a Google Doc. If you want to like make sure you put your screen name in and like type a short question for Dr. Garte. Now there's a lot obviously in the paper we haven't gotten into, but I really thought the final paragraph was very interesting and had implications on a variety of fronts. And you've kind of been talking about this, right? So quote, however, it should be noted that no assumptions were made on the relationship between cellular complexity and survivability, while the results suggest that low survival probability precludes evolution in a continuous stepwise manner. It says nothing about evolution from simple to more complex early life forms, assuming that biochemical complexity is not tightly linked to survival probability. There is, in fact, no a priori reason to believe that simple life forms could not have high innate survivability. In that case, further evolution of simple forms would not be possible even if initial replication fidelity is low. Now, when I read this, all sorts of questions popped to mind. We've kind of been discussing different aspects of it throughout our conversation, but can you expand on this and kind of culminate everything we'll be talking about in terms of the relevance to the most predominant origin of life theories and then obviously in this kind of evolution, like the standard evolutionary models in general, if that makes sense?
Okay. This... So what I'm saying here is that this is not connected to complexity. This idea, an evolution of complexity is a whole nother topic which I don't talk about. But it could be possible that a simple life form could arise with initially very high innate survivability. I mean, you can't rule that out a priori. I think it's a very, very low likelihood that that's true. You can't imagine. I can't imagine a scenario where that would be true. On the other hand, and you asked about abiogenesis theories, and my answer to that is, what theories? I don't know one. I really don't know. I remember, a theory has to take into account all the facts that are known. There is no such theory in abiogenesis, and it doesn't matter what you're dealing with. It could be, production of biomolecules. It could be concentration of biomolecules within a membrane. It could be synthesis of biopolymers. It could be information. It could be systems development and how they all work together, as we've been discussing. There's no theory for any one of those, not to mention all of them together. And I had a comment on my debate last night on Modern Day Debate. Somebody commented later saying, "Oh, he's full of that's nonsense. Abiogenesis is coming out with new papers every week that are pushing the field forward." And I answer simply saying, "Could you send me one of those?"
Well, it's interesting, Dr. Garte. I'm wondering how long it will be. I'm sure you've gotten this in the past, but I'd be interested to see how long it will be until the standard response to your arguments is the same one that Dr. Tor gets.
Oh, it is already.
Yeah. Oh, well, you just don't understand the chemistry, and that always cracks me up in a lot of these conversations. I'm like, people don't seem to grasp the point that it's you understand the chemistry and the arguments that are being made. You're questioning the plausibility on a variety of fronts, and hey, here's the evidence that says, from a experimental perspective, that says this is not plausible, or this doesn't work, or we need to account for X, or both. All of these things would have to be accounted for, so on and so forth. But just because your conclusion is different than somebody else's doesn't mean you don't understand the foundational concepts. And it blows me away how folks like you just get like, "Oh no, he doesn't understand it." And it's like, well, okay, you can make that argument towards me. Okay, I don't have a PhD in biochemistry, haven't read a bunch of papers, don't have an H index to even like look at, let alone have one that's successful. But it doesn't mean that I don't understand the concepts, but more importantly, that folks like you, just because you don't agree means that you don't understand it. And that's what always fascinates me. But it's like, are we actually having intellectual discourse, and are people actually trying to reach conclusions or have actually rational critical thinking as they analyze these things, or are people only looking for things that fit their particular worldview? I don't know what you think about that, but it seems to me that's what's happening.
For sure. I mean, I'm nothing like Jim Stump. Jim Tour. Jim Stump is another friend of mine. Jim Tour is probably one of the greatest chemists alive. And, when Professor Dave, as you mentioned, says, "Well, Tour doesn't understand," I just laugh. I mean, Tour doesn't understand. What a lot of people who are not scientists don't realize is that you can specialize in something, and Tour specializes in nanochemistry. But when you work in a field for a long time, any field, you're so involved in it that you specialize in it. But at the same time, you're picking up a huge amount of information from all the other fields that surround it. And so, I am not a abiogenesis scientist. I won't show up in the list of the top 100 abiogenesis scientists, but that doesn't mean I don't understand it. And I'll tell you the truth, if you got Jack Szostak or John Sutherland or Jerry Joyce, or always leave out a few people, if they're listening, which I doubt, I apologize. But if you got all those people, Eugene Koonin is another one. If you get all those people together and ask them in a bar, what do you think about the field? They'll say exactly what I'm saying. And exactly what Jim is saying. This is really hard. We're not doing too well. We got to work harder. And I say bless them, because they are working hard. and they're trying their best. And I don't think it's going to help, because the problem is not amenable. And this is where I really come down, because I didn't say this in the paper, but this is what I believe. It's going to come down to changing our entire paradigm when we look at abiogenesis. It's not reducible to chemistry. That's a tough sell. Nobody wants to hear that. Biology is not reducible to chemistry, so why should abiogenesis be reducible to chemistry?
Right. Well, in line with that, one of the things that I forget which paper it is now, but there was a statement that was made. This was in relation to like the genetic code, and it was basically, I'm paraphrasing here, but it was the experimental evidence now shows that the genetic code operates within the laws of physics and chemistry, but is not dictated by them.
There you go. And
I think, correct me if I'm wrong, but it seems like that you're saying that same concept applies to abiogenesis.
Absolutely.
Yes, it can operate inside of course it operates inside of the laws of physics and chemistry. That's a given, right? But it doesn't mean that it would be caused purely by them. And
the way I would put it, and again, this for some reason is controversial, I don't know why it should be, is that biology is definitely an emergence of chemistry, but it has its own laws that don't apply anywhere else. It has its own scientific, quantitative, maybe biological laws, which we don't know. We don't know what they are. We only know a few, and they're very, very small. And I don't think we can get to understand those laws with the metaphysical naturalistic approach that we use to do science. We're going to have to expand it somehow. And that could include a creation model. It might not. It might include something that's related to creation. I like to talk about teleology, and I've recently been talking about agency as things that have to get back into biology, because all biological organisms are full of purpose. I mean, we're only alive because of teleology. Okay, if we didn't feel hunger pangs when our blood sugar is low and we haven't eaten for a while, we would die of starvation. It's the hunger pangs that make us eat.
Right.
And that's, that is a purpose. Every enzyme has a purpose. We call it a function, but it means it's a purpose. The purpose of, amino acyl tRNA synthetase is to link together the correct tRNA and the correct amino acid, which has a anticodon on it, and then that enzyme puts them together. It makes sure it got the right one because it has an editing function, an error corrections function, and then it activates it with. I mean, it's an amazing molecule. This is a chemical molecule that does more stuff than we can do as human beings. We can't do that. It's a molecule.
To me, when you really go down the proverbial rabbit hole and look at the different things, the things you're describing, right? And what you're describing, while it's an extraordinarily complex component of biological systems,
right?
There's just almost an endless plethora of ones that we're discovering that are almost every bit is like, what? I mean, I remember the first time I saw an animated model of like ATP synthase.
Oh yeah.
And I just like, okay, well, that's number one, that's very awesome. But number two, it's just kind of awesome. That's just a mind bender and then the part that always comes back to me is you ultimately have to circle back around to both the purposeful function that you were referring to, but like this three-dimensional object, i.e. the molecule, is the direct result of digital information, which houses the information necessary for the synthesis of the three-dimensional object.
Right.
And to me, for me personally, that's where a lot of the questions about like, what is the inference of the best explanation in terms of the root causes that are necessary? Like we have these gigantic, they appear very almost insurmountable barriers on the chemistry side of creating the different components that are necessary. But then the organization of said components into digital information housing molecules which like being done, and this again goes back to your whole point about your paper of like the fidelity, right? At some point, you still have at some point, whether in your paper you're not addressing it, but at some point in this process, you have to get to the point where you're having to account for the fidelity of the replication of digital information.
Exactly.
And to me, when you contemplate that, and it gets into the aspects of like the error correction mechanisms, all this kind of stuff, like when we have direct equivalent of Hamming error correction codes, like that are operating inside a biological system, to me, it's like, which one requires the greater leap of faith in terms of what would be necessary from an origination perspective and a direction standpoint and the formation of these necessary functional components? Even if you want to argue that if you have all these components accounted for, then theoretically you could account for the expansion from almost like an AI, artificial intelligence perspective and controlling all the algorithmic improvements. Even if you want to account for that, getting to that point, people don't seem to want to account for the amount of coherent functional information that would be necessary in order to accomplish those root functions. And I think Shostak had a very interesting paper on where they were defining, the functional information, how you quantify it in DNA. And it's a very, very fascinating paper. But then going back to, one of you and I's, favorite topics of all the folks who claim it's not even a code. And it's not really information, right? I'm like, we have PhD, Nobel Prize winners who are sitting here over like coming up with formulas of how you quantify functional information in a gene sequence. And then on the flip side, we've got folks from a worldview standpoint who are denying that that's even something to be accounted for. And to me, that's it showcases the how broad the gap is from between like the truly analyzing things, like you said. Many that you get in a room with a lot of these origin of life researchers, they'll freely tell you, well, we're not making too much progress, right? And on the flip side, you've got the people you referred to, like, no, they're publishing new stuff every week. We're getting so much closer, right? And it's like a polar opposite of actual reality. I don't know, am I correct in that, or am I just like getting a little bit jaded with all these YouTube Evo agents?
Oh no, you're absolutely right. Absolutely right. Can't agree more.
Well, Dr. I appreciate you spending so much time with us. We got a couple quick questions from you. Slammer's got one for you, and Rebecca, I've got one for you, and then we'll wrap up. Folks, if you have any questions for Dr. Garte, we're keeping for like maybe 5 or 6 more minutes, or get over to, I'll post a link to the Google Doc because I know folks are having problems with the spreadsheet. I'm going to post that link in chat real quick, make them short and sweet. And here's a link to, oops, that's not what it is. And we'll get these and let Dr. Garte get back to recovery from his debate last night. And, all right. Okay. So Rebecca said, by the way, it's Bread of Life. Awesome channel. Go check her out. Rebecca, make sure you post a link into your channel in the side chat. But she says, why do origin of life researchers like Dr. Kunev, I think that's Dimitri, speak so confidently that they have all the steps for origin of life figured out? Are most origin of life researchers so confident, or do they recognize the problems you were discussing? I think we kind of touched on that, but I know Dr. Dimitri, so maybe allude to
that. Eugene Koonin, is that is it Eugene Koonin? I don't know any.
No, Dimitri.
Dimitri. No, I don't know what she's talking about.
I think that's his last name, isn't it?
Dmitry Kunev. Yes.
Dmitry,
yes. I know him well. Why does he say that? Okay. I know Dmitry quite well. He's a good scientist. He's working on some interesting issues with the origin of life. And he knows, I will say this, I mean, I've talked to him in great detail about this. He knows that it's a real problem to understand how, for example, what he's working on is how the genetic code evolved. What are the early steps in how the genetic code came to be? And he understands how difficult that is. He's looking at a particular hypothesis, which involves the peptide RNA world. He's up to date on that. And I think his statement, which I heard him make on your channel, Rebecca, that we are making a lot of progress, we're getting closer and closer, is his philosophy and not really coming from his science. He may disagree with me, but I think that philosophically he feels that there are, I mean, I don't want to talk to him for him, but I think that what he thinks is that philosophically, since there have been some advances in this field of peptide RNA world, I personally consider them to be extremely small advances. They mostly relate to chemistry. He's pursuing a chemical evolutionary theory for the origin of protein synthesis, and I think it will only go so far. There's a point at which it's just going to stop, because it's only the very beginning part that he's looking at. And I think what you said, John, comes into play here. You can always publish a paper, and many people do, with a very, very small quote advance, which actually is just testing a small aspect of a small part of a small category of one small aspect of abiogenesis. And you can do that a lot. You can do that, there can be a million of those papers, but when you add up all those millions, you're still going to have a very tiny fraction of the questions answered. So I wish Demeter all the best in his work. I hope he actually is able to come up with something which he can then publish, and I think he will. But I don't think it's going to advance us to the point where we can say, oh, it's becoming clear. No, we've got a long way for that.
Well, one of the things that I'm not up to date on all of his stuff, I read some of his stuff, but one of the trends I seem to notice in all these models for evolution of the genetic code, they all seem to boil down to like, oh, as long as we don't have to account for the whole thing all at one time, and over, eons of time, we rant ourselves to have all these chances to somehow, oh, well, we only need to account for this number of codons and these amino acids for like this period. And then like in step number 2 or stage 2, we're going to get this next 4. And then stage 3, we're going to get these. Like, to me, I'm like, I kind of question the plausibility of that.
I guess let me give an analogy. I don't know if it's a great one. I just thought of it. But let's say you have a kid in school who's having trouble with math. Okay, it's really having a hard time with the multiplication tables, and you drill him. You keep having him do exercises, and one day he comes to you and says, you get testing him, and you say, what is 7 times 3? And he says 21, and he got it. And you and your wife say, yay! Tomorrow he's going to solve the Schrodinger equation for for helium or for carbon. No, it's great that he learned 7 times 3, but now he has to understand 7 times 4 and 6 times 8, and finally he'll get it all. And then he's got multiplication. Does that mean he's now a mathematician? Not quite. Progress is great, but this kind of progress doesn't mean you're ready to understand exactly Gödel's theorem. I mean, you've got a long way to go.
Well, to expand your analogy there, I almost feel like in your paper, you're not talking about the complexity necessarily, but obviously at some point in abiogenesis research, you do have to address the complexity.
Oh, absolutely.
And I kind of feel like in some ways I'm not talking from an arrogant perspective to understand how complex everything is, although I recognize like, holy crap, this is like it's so complex, we really don't even comprehend how big the problem is in its totality. But I almost feel like to some extent, a lot of the origin of life researcher folks they're the kid who just figured out how to solve 3 times 7. Exactly. And then they now think they're going to solve Schrodinger's equation, et cetera, et cetera. And I'm going to figure out the solution to the universal wave function. Something to that effect, right? Like, something I thought about was I recently read for the second time, I read Meyer's Return of the God Hypothesis.
Right,
book.
right. Good book.
And the portion, he spends a good portion at the end of the book actually diving into the details of the universal wave function problem for the universe. And it's, to me, I think almost like there's like the equivalent from a abiogenesis perspective. We almost have a universal wave function for biology that has to be accounted for.
Yeah, I agree.
As well as on the cosmology perspective, like you have to count for both of them in that context. And anyway, it's a mind-bending stuff. Okay, one more question, and then we'll wrap up. So the one and only Slam RN, thanks so much for joining us. She says, have you seen the latest that the Chinese redstone Cambrian layers show that the entire Cambrian explosion took only about 410,000 years? Is this a problem for evolution? Then says Cambrian explosion now dated to no earlier than 538 plus or minus 0.21 million years ago to no later than 538.58 plus or minus 19. So wow, that's crazy, because I've heard many times, there's millions of years to work with. And now she's saying that it looks like there's only about 410,000 years to work with. What are your thoughts on that?
Well, I haven't seen it. I think that the Cambrian explosion, no matter how long it took, is an example of discontinuity in evolution. In fact, that's one of the examples. I think maybe I didn't use it in the paper, but I used it in other things. I used the origin of vertebrates, the origin of, and this would be the origin of various phyla. And how long should that take? I have no idea. I don't know what. I've seen that I've seen that result, Susan, in the popular press. I haven't read the original paper, so I don't. I can't comment on it, and also it's way out of my field. But I think originally it was considered to be about 10 million years. Well, if it's really a 20th of that time, I don't think it disproves evolution. I think it proves my point that discontinuity is an important factor. I mean, the origin of heterozygous, if it's true, which everybody seems to think it is, and I don't have any reason to doubt it, could have happened in one generation. I mean, it was one large prokaryotic cell that engulfed, a blue-green a cyanobacterium. That took a few hours, okay, and it changed the world. It changed the universe, because that was the beginning of eukaryotic cells. However that happened, nobody knows how it happened. But if it did happen, and there's no other alternative hypothesis that I know of, it happened very, very quickly. The whole gene duplication that occurred starting vertebrates happened in one animal, possibly in one fish, or pseudofish, and the next thing you know, we get vertebrates. So that's the thing about discontinuity; it can happen very, very fast, and I think it's something that has to be taken into account.
Well, something that to me observing what we are starting to be able to do on a very simplistic level in our own attempts to reverse engineer and re-engineer different organisms for new functions, right? To me, when you look at some of these conundrums, they are no longer conundrums if you insert an intelligent agent into the equation.
Yeah, that's right.
And it's like, so let's just hypothetically say that old Earth is correct. There is these different stages, and then we have a Cambrian explosion that is not, that's not just evolutionary caused by evolution from the traditional sense. Well, I mean, it's like, well, okay, so the intelligent agent that programmed everything originally anyway comes in and does a download, inserts additional body plan information or whatever into different organisms, and presto, the next generations are going to be able to express these new phenotypes. And I'm like, that's not that big of a leap, right? If you're inserting an intelligent agent into the equation versus having to somehow account for literally billions of mutations that would have to take place and be conserved in order to account for like entire new body plan formation and all these different phyla and all this stuff that comes into place. Like it almost seems to me like it's one of those things that people don't really want to address because of just the staggering amount you'd have to account for in order for it to happen through just like a traditional evolutionary process. That's what my take on. I could be wrong, but that's kind of like the way I look at it. Of, hey, we toss in the intelligent agent into the equation. hey, we're talking about like doing an upload, right? Like we install new stuff on our computers all the time. Like this is not that big of a leap.
Well, look, if we're really talking about what I think happened, what I believe happened, we have to leave science, because I believe God created everything, and God created life as well as the universe. And I believe God created humans, because we are not, I don't think we're the product of evolution either. So that's what I believe, but that's not science. That's faith. And it's good to have science to back up one's faith. And I do. I have plenty of science to back up my faith. But, in terms of what actually happened that we can address in a naturalistic way, we don't know. Now, that I agree that there's a lot of, there are pointers. The strongest pointer is to the origin of the universe. That points to God. There's no alternative, or we don't know. There isn't an alternative hypothesis. For life, there's not much of an alternative either. As I said, there are no theories that really are, at this point, that are good. They may eventually come some, but my prediction is that if we ever get a naturalistic theory that is good at making some progress towards understanding the origin of life, that theory will have to include one or more aspects that point directly to God. And I don't know what those are. I have no idea. But that's what happened in physics.
Right.
Okay, physics was stuck in, as I said last night, in 2004. And the only way it got unstuck was to include things which at the time were mystical. Okay, time speeds up, slows down, that's mysticism, right? That would have been called mysticism in 2004. The whole quantum theory was mystical in 2004, but suddenly it became science. So that's going to happen in biology now. We're there. We're at that point. If we want to make any progress, we're going to I talk about teleology and agency, but I don't know if that's right. It may be something else. It may be something, but whatever it's going to be, it's going to point to God, just as, in my view, modern physics points to God.
So are you saying that it might boil down to Aquinas was correct, and that ultimately it boils down to a prime mover? What the prime mover moved could be subject to discovery, but ultimately at some point you would require the prime mover to be in place, even if there were naturalistic mechanisms that were enabled these other things to account for. You still have to have and that's why, in my opinion, when you look, I was actually having a conversation with a guy earlier today about this, and we kind of got off on a sidebar about like dualism, idealism, and materialism. And the kind of point that I make, and I stand strongly behind what I'm about to say. I feel that strict materialism forces you to suspend, at a minimum, 50% of your awareness and understanding, or potential understanding of your existence. And
probably right.
And more than likely, it's probably more than 50%.
Oh, I think it's more. Yeah, I do.
But I'm saying at a minimum, it would be 50%. And I don't know. I almost feel sorry for people that find themselves so contained in that worldview, because, for me, when I'm walking around and just contemplating these things, I'm truly in awe of just the astonishing aspects of it. And then beyond just what it does and the functionality, into the mind that must be behind it ultimately. And that transcendence is astonishing. And then the premise of, at some point when I shuffle off the mortal coil, to be able to engage in that plane of existence, to me, that's like a mind-blowing, fascinating, like, all right, well, that's pretty cool. And then the concept of, this is all you got, and make the most of it while you're here. And I'm like, I'm trying to make the most of it while I'm here too. Like, I'm not like ignoring what's going on in this plane of existence, but I've got something to be pretty excited about from the macro view of my, eternal existence. And the concept of so many people who like, oh, I wouldn't want to be able to like exist. I'm like, Okay, that seems a little bit strange. Are you not curious? Like, do you have no curiosity about like all the different facets of existence? I mean, I don't know. We can talk about that forever. Dr. Garte, thank you so much for spending your time. Appreciate everybody who joined us. I know I have been out of the YouTube game for the past like month and a half. It's been kind of crazy winter season, or I guess fall, Christmas season. But I am back, so hopefully I will be back here on a regular basis. Thanks so much for joining us. Dr. Garte, thank you as always for sharing your knowledge and time. And folks, as I always say in these conversations, as you contemplate the different aspects of your existence, all of the variety of, just concepts that we must address in relation to our very existence, make sure that whatever conclusions you reach are logical, plausible, and probable. Have a good one, folks. Appreciate you so much.